All right, hello everyone. So the protein that I had was the Piezo1 protein with the mutation Arginine that is replaced by histidine R2456H, and the conditions were malaria resistance and hereditary xerocytosis. So here is my protein. That's the channel, these are the beams, these are the blades, and yeah, we're just going to talk about it. Okay, so the normal function of the Piezo1 protein is helping with detection of the interspace or interluminal pressure changes and urine flow sensing. I would categorize this protein as a transport protein because it allows calcium to flow through the channel, this purple part here, it goes through here, and while also monitoring the flow. And the protein's activity is activated when positive pressure is applied to the resting tension caused by the membrane or the cytoskeleton, depending on where the protein is. And it needs applied pressure to activate and send calcium ions through the channel. And this process could overall be called cation transportation. So the protein is normally found embedded in the plasma membrane in the cells of the kidney, bladder, and blood vessels, and are usually found in eukaryotic cells in animals or humans. The type of change that takes place, I would call it a substitutional change because arginine is replaced or subbed out for histidine, and the protein becomes more active because the inactivation is slowed. In other words, they kind of cancel out or it becomes a double negative. And the fact that inactivation or the causing of the channel to close is slowed down, meaning the rate of calcium going through is a lot higher. And I also wouldn't describe it as a structural change because the overall structure of the protein does not actually change. The overall structure doesn't change. It's just this part is affected because it doesn't move as quick as it should. And so this is my model. It is made out of play foam and hot glue. It does not depict the mutation in a way that is obvious and wouldn't require an explanation because, like I said, the mutation isn't actually something you can really see overall. You'd have to understand that it literally just takes place in these areas. And yeah, the mutation affects the channel right here, these purple parts, and then the beams, the green parts. Yeah, and because of the mutation, the beams are slower to close. So usually they would open, calcium ions would go in, they'd close, but because of the mutation, they close a lot slower, so it's just letting in more calcium that's not needed. So for the main functional group in the original side chain of arginine, it is a complex side chain of mostly hydrogens, carbons, and an amine group because of the nitrogens. And the side chain is considered one of the most hydrophilic amino acids because it includes a large quantity of guanidine and can make up to 6 hydrogen bonds. Arginine is polar covalent and positively charged. This amino acid is one of the bigger ones. And because arginine is hydrophilic, it can interact strongly with water and form hydrogen bonds with other hydrophilic side chains. And arginine can also form ionic bonds because it has a positive charge. And the mutation requires that arginine be replaced with histidine. Histidine is very similar to arginine and has a side chain of mostly carbons, hydrogens, and it has a carbon ring with nitrogen, making it have an amine group. So like arginine, histidine is polar covalent and positive, and it is only slightly bigger than arginine because it has the ring. And histidine can also form hydrogen bonds and ionic bonds because it's polar covalent and positive. Also, histidine can exist as positive or negative depending on the pH balance, the pH range. And when comparing the two, they're fairly similar in the fact that they're both, again, polar covalent and positive. However, the ring structure on histidine makes it less flexible than arginine, which could account to why the beams are slower to close because they're not as movable, I guess you could say. And then replacing arginine with histidine leads to the Piezo1 protein being slower to react, like we've talked about, to pressure change and close its channel, and this leads to overactivity in the protein, allowing excess calcium into the channel and then into the blood cells. And the cell can then become dehydrated and gain hemolytic anemia, and this causes the organism or the human to experience fatigue, jaundice, and darkened urine. In relation to malaria, however, it's beneficial because, like I said, the cells become dehydrated, which causes them to become resistant to the malaria parasite. So having the mutation is actually very beneficial in third world countries or in places where malaria is a lot more rampant. And to lastly evaluate the model, so like I said, my model was hot glued together, so I'm not able to completely demonstrate the effects of the mutation. If I used a more flexible or stronger material, I might have been able to get the beams to move open and closed. Also, the model does not depict the channel exactly as it appears on the actual protein because the foam is a lot more fragile than anticipated. Like the channel is on the actual protein is a lot longer, and it doesn't completely connect on both sides. It connects on this side alone. It doesn't really connect here. So, but in order to keep the protein together, I had to connect them both. And then also the channel was harder to model and show than it is seen on the actual protein, like I said. And then the blades are not as uniform or as flat on the model as they are on the actual protein. Like the blades are usually on here, if you can't tell, they're round and fat, but on the actual protein they're flatter and curved, and they're probably about the on the picture model, they're about as, wide as my hand, and they're very flat and they're curved. And it kind of looks like one of those star blades that you throw. But yeah. And that is all for my protein.